Fibrodysplasia ossificans progressiva is a rare musculoskeletal condition where, after birth and progressively through life, muscles and tendons are gradually transformed into bone. There is currently no cure for this condition, and with proper care, a person's life expectancy is usually around 40 years. Fatal familial insomnia is another devastating condition characterized by a complete inability to sleep that is unaffected by even the strongest sleeping pills. Symptoms include constant panic attacks, hallucinations, and drastic weight loss, eventually leading to a state where the patient is unresponsive to their surroundings. Death usually occurs within 18 months, and there is no effective treatment. Progeria is a rare genetic disorder that causes premature aging that begins in infancy. Despite attempts at treatment, there is no satisfactory cure yet, and children suffering from the disease usually do not live to the age of 15. Although these diseases are extremely rare, the consequences for those who suffer from them are profound. Patients and advocates emphasize the urgent need to prevent even one person in a million from developing these diseases. More than 6,000 genetic diseases are currently known, and new ones are constantly appearing in the medical literature. Do you know how many of them are treatable? 600 out of 6,000. 24% of people in the world are carriers of at least one genetic disease. Obviously, the solution to the problem is to eliminate the cause by gene editing. However, the inclusion of genome editing in the 2016 World Threat Assessment of the U.S. intelligence community was a historic moment, equating it to weapons of mass destruction, such as nuclear missiles and chemical weapons. Why? In 2012, the advent of CRISPR-Cas9 genome editing technology revolutionized biotechnology by making it possible for almost anyone to modify and delete segments of DNA inexpensively. The straightforward answer to the question, why, is that bioweapons development has evolved from a resource-intensive activity requiring significant research and production capacity to the rapid, efficient, and undetectable creation of pathogens suitable for mass casualties using synthetic biology. The U.S. National Academy of Sciences produced a 221-page report on the implications of CRISPR for the U.S. military in an effort to provide a comprehensive assessment of its potential threats. Prior to the report, there were differing opinions, prompting the Pentagon to ask the entire U.S. Academy of Sciences for a consolidated assessment. So what did the scientists cite as an example? For example, the creation of artificial viruses, such as smallpox. Ridiculous. They claim that experiments are already underway to create deadly viruses for which there is no vaccine, and so on. However, this is all wrong. What other viruses and E. coli? We are talking about the dangers of gene editing, the natural code that makes all living things what they are. Let's explore the question of the dangers of gene editing from a greater distance. Look at what you're experiencing. We derive most of our pleasure from aesthetic perception, particularly from contemplating beautiful faces. But what defines beauty? While this concept is often considered subjective and varies by age, ethnicity, and culture, scientific studies reveal a surprising consensus across any group regarding facial attractiveness. A beautiful face is a universal category. Francis Galton is famous for developing the composite portrait method. Who has not seen the video? A new era of humanity has begun. I remind you, to obtain a composite portrait, he superimposed photos of the faces of criminals on each other, getting an average image of the whole sample. Galton noticed a striking phenomenon. The resulting image was more attractive than any individual face in the group. This raises an intriguing question. What accounts for this collective attractiveness? Our faces play a crucial role as indicators. We have learned in the process of evolution to estimate the external attractiveness of a person in 1/10 of a second thanks to ancient brain structures. And then we think to ourselves that we like a person, for example, because of his or her competence and charisma. A beautiful face is an indicator. But an indicator of what? Why do we find certain faces beautiful? One plausible hypothesis is that attractive facial features correlate with genes that have proven useful during evolution. When we look at a composite image of human faces, it is as if we see a preferred middle ground that has been tested for thousands of years, sharp deviations from which are dangerous, as in target shooting, when it is advantageous and simultaneously safest to aim at the very center. More broadly, attractiveness offers many benefits. Attractive people tend to be more trusted, perceived as competent, and their positive attributes are more quickly noticed. They often earn higher incomes, and studies show that they receive more care and attention from parents from an early age. Beautiful people find it much easier to succeed in life. This begs the question: have you ever regretted aspects of your appearance that you are unable to change? For genetic reasons, it is unlikely that most of us will ever find ourselves walking the runway as supermodels, for the same reason that there are no 150-centimeter players in the NBA. While motivational videos, books, and movies often emphasize hard work and determination, reality emphasizes the role of genetic advantages. For example, Eero Mäntyranta is a legendary skier in the history of the sport. Between 1960 and 1972, he managed to win 7 Olympic medals, including 3 gold medals and 2 world championships. However, his success owed much to a rare genetic mutation that increased hemoglobin production, which greatly increased his endurance and performance, as detailed in David Epstein's book, The Sports Gene. Success in sports is multifaceted, influenced by factors such as nutrition, coaching, access to opportunities, and even luck. However, certain genes can actually play a crucial role. One such gene is ACTN3, known for its effect on the speed of muscle contraction. Despite extensive gene research, ACTN3 is still unequivocally associated with a predisposition to strength sports. Studies involving mice in which the ACTN3 gene was turned off showed a significant decrease in muscle strength. Although having a favorable variant of ACTN3 does not guarantee Olympic success, people with 2 non-functional copies of ACTN3 have virtually zero chance in sprinting events. Conversely, if you have a mutation in the MSTN gene, which regulates myostatin, a protein that inhibits muscle tissue formation, your muscles will grow many times faster than others. You heard me. Your own body prevents you from forming the perfect silhouette because muscle is a very resource-intensive thing. Evolution doses this sort of thing out. For example, in a 2004 study in Germany, a notable case was a child born with a mutation in the MSTN gene. Note the muscular development of the 7-month-old baby. This condition allowed the child to exhibit strength far beyond that of his peers, demonstrating the profound impact of genetic variation on physical capabilities. A similar case can be seen in American toddler Liam Hoekstra, who learned to do pull-ups at just 8 months of age. Animal studies show that inhibiting myostatin leads to a significant increase in lean muscle mass with minimal fat accumulation. Mice, dogs, and cows with mutations in the myostatin gene provide impressive examples of this phenomenon. Wouldn't you like to acquire a similar mutation, especially now that calories are in abundance? Isn't it a dream to build muscle while doing nothing? While exercise and a balanced diet can improve cognitive abilities, your response norm sets an upper limit on achievements such as muscle growth or memory improvement. This norm represents the potential dictated by your genes. You can optimize your performance within this range, but you cannot go beyond it. So while lifestyle choices can optimize your genetic potential, they cannot fundamentally change the genetic constraints that determine your capabilities. As American psychologist and geneticist Robert Plomin said, if a child has a weak memory, it is likely that it will remain weak. No matter how hard teachers and parents fight, he will not grow up to be the world's best mathematician. And if genes mean so much in human life, genome editing, at least in the long run, is inevitable. And not just in the case of inherited diseases. Do you want your child to grow up smart? Isn't that so? Improving intelligence, often measured by IQ tests, remains a challenge. IQ scores tend to remain stable throughout a person's life, and the older you get, the more IQ becomes a genetic trait. Many of our cognitive abilities that proved useful to our ancestors, memory, pattern recognition, language, and math skills, are interrelated. Thus, people who excel in one cognitive domain often perform well in others as well, which is the basis for measuring intelligence. The true meaning, however, lies beyond cognitive tests. Numerous studies show IQ's relationship to health, educational attainment, career success, and even longevity. Not surprisingly, above-average intelligence often correlates with attaining high-paying positions, but this result is not guaranteed in every case. People often underestimate how much nature influences their abilities and behavior. They may dismiss other people's difficulties by saying, "You're just lazy. You lack patience. Get a grip." While such statements may be fair in some cases, many people don't realize the difficulties faced by people with significantly lower IQs. There are some people who work tirelessly despite these difficulties. Tasks that are easy for others can be extremely difficult for them. It is important to recognize that genetics plays a significant role. Our biological makeup is the culmination of 4 billion years of evolutionary history. Throughout this vast period of time, evolution has operated according to uniform principles. But now, as Jamie Metzl argues in his book, Hacking Darwin, Genetic Engineering and the Future of Humanity, the rules may change. This video is inspired by that very book. Who is this Jamie Metzl, you ask? Jamie Metzl is a technology futurist, American author, and senior fellow at the Atlantic Council of NATO. In his book, Metzl, who has experience at the National Security Council, the U.S. Department of State, and various other positions, explores predictions of humanity's future development. Predicting the future of humanity may seem absurd, but history suggests otherwise. In 1962, at the height of the Cold War, U.S. President John F. Kennedy boldly declared that the United States would land a man on the moon before the end of the decade. Despite the risk to America's credibility, Kennedy's idea relied on existing technology such as rockets, heat shields, life support systems, and computers to do the math. His statement was not mere speculation, but a logical extension of existing capabilities with the necessary minor adjustments. Almost everything was already in place. The inevitability of the event was obvious, and 7 years later, this happened. The genetic transformation of our species is also now moving from speculative fiction to tangible reality, a fact that may not have been widely known until now. This gap in public awareness and scientific progress emphasizes that public understanding of science is evolving linearly, while science itself is evolving exponentially. According to Jamie Metzl, the future developments you are about to learn about are inevitable because all the necessary tools are already in place. The only question is whether these changes will take place in the coming decades. What is certain is that within a century, the traditional way of having children in developed countries is likely to be obsolete. So, does this woman seem unnatural or even unnatural to you? Her name is Louise Brown. She was born on July 25, 1978, in England and became world famous as the baby of the century. A poll conducted shortly after her birth showed that 93% of Americans knew she existed. But who is Louise Brown? She holds the honor of being the world's first child, conceived through in vitro fertilization, IVF. IVF, a technique used when natural conception proves difficult, was revolutionary but controversial, especially in its early years. While many note that the technology allowed infertile couples to have children, not everyone shared this view, especially during the time of Louise Brown. Her parents received threatening letters in packages with plastic embryos stained with blood. Catholic theologians condemned IVF as a moral outrage and considered the process of creating children in vitro unnatural. People often have a rather strange idea of what naturalness means. They advocate using only natural products and living in harmony with nature, without fully understanding the origin of the products they consume. Do you know what that is? This is corn 9,000 years ago before agriculture was invented. There's the carrot, the eggplant, the watermelon, the banana. These fruits and vegetables, even those labeled as organic in fancy stores, are the product of deliberate breeding over millennia. Is it natural? Agriculture itself can be seen as a global genetic modification initiative. Soviet biologist and plant breeder Ivan Mikurin articulated this view. We cannot wait for favors from nature. It is our task to take them from her. Imagine you are faced with a severe bacterial infection. What would you choose to be treated as you were tens of thousands of years ago, with berries and rituals, or antibiotics that could save your life? Our idea of what is considered natural is deeply rooted, but much in this world is far from it. Over the millennia, humanity has been actively changing our environment. Why? Because nature, despite its origins, has always posed a threat to our survival. If you were to travel back 100,000 years, you would encounter many creatures that could end your life very quickly. On the physical side, humans are very weak animals. However, our modern tools and weapons have elevated us to super predators on Earth. The safety and comfort of our modern environment is largely due to our collective rejection of all things natural. Since the birth of Louise Brown in 1978, despite initial protests and skepticism, more than 8 million children around the world have been born through in vitro fertilization. Remarkably, these IVF-conceived children have been found to have no more physical abnormalities than their naturally conceived peers. In the United States, about 1.5% of all babies are born through IVF, and in Japan, the figure reaches about 5%. The reason to pay attention to this is obvious. IVF remains a complicated, painful, and expensive procedure with varying degrees of success. However, continued progress suggests that this may change in the future. Once this situation changes, those parents who have children in the standard way, rather than through IVF, will be looked upon with the strongest condemnation, condemning their irresponsibility, similar to how parents who do not vaccinate their children are treated today. In 1990, 12 years after the historic birth of Louise Brown, doctors made a breakthrough by successfully testing preimplantation human embryos for genetic diseases, a procedure known as preimplantation genetic diagnosis, PGD. In the IVF process, where multiple embryos are usually produced, PGD allows for the identification and selection of embryos without obvious genetic diseases prior to implantation. The benefits of PGD are obvious, but ethical questions arise. Where do we draw the line between eliminating disease and enhancing desired physical attributes? If we can select embryos free of diseases such as muscular dystrophy, what prevents us from selecting embryos with a genetic predisposition to increase muscle mass? And we're definitely one step away from crossing that already extremely blurred line. And someone has already crossed it. Here are the websites of clinics in the United States that allow you to undergo IVF with screening right now, not only to rule out certain genetic abnormalities, but also to simply choose the sex of your future child. Advancements in reproductive technology offer increasingly diverse and sometimes controversial options. For instance, some clinics now allow parents with conditions like dwarfism or deafness to select embryos with genetic traits associated with these conditions. The ethical implications of such choices are complex and contentious, raising questions about the boundaries of genetic selection. Consider Down syndrome, where individuals are born with an extra copy of the 21st chromosome, leading to developmental challenges, cognitive disabilities, and increased health risks, including heart defects and cancer. Despite these challenges, many individuals with Down syndrome lead fulfilling lives, contributing positively to their communities and society at large. For families and friends, they are often seen as blessings. Interestingly, since the 2000s, Icelandic doctors have been mandated to inform expectant mothers about publicly funded screening tests capable of detecting Down syndrome and other genetic conditions with high accuracy. During 10 years of such tests, Almost all women who tested positive for their child's Down syndrome had an abortion. Australia, China, Denmark, the UK are all the same, with abortion rates as high as 90 to 98% everywhere. Even in the US, where abortion rights are surrounded by an ongoing religious and ethical debate, a 2007 poll found that only 20% of Americans believe that parents should have the right to terminate a pregnancy because of a serious but not fatal genetic abnormality such as Down syndrome. Despite this, 67% of Americans have made the decision to have an abortion after receiving a Down syndrome diagnosis. This gap highlights the stark contrast between moral beliefs and actual decisions, especially when it comes to a choice as difficult as abortion. Looking to the future, imagine the consequences when parents can simply choose which of 15 embryos to implant. This underscores that the discourse of high moral values is often at odds with the decisions people end up making when faced with complex ethical dilemmas. If we justify screening out embryos that may produce children with Down syndrome because of potential unhappiness, why don't we select embryos with genes that can maximize happiness? As we said, studies show that the higher the IQ, the higher the life expectancy, income, and happiness. It's only natural for parents to desire these benefits for their children. But what level of IQ is considered high, and how do you achieve it? To get to the bottom of this, let's start with the chicken. Gallus gallus, the ancestor of our domestic chicken, originally laid only 1 egg per month. Through centuries of breeding, humans have evolved them into modern chickens capable of laying up to 300 eggs a year, 25 times more. The point is that hacking biology is much easier than it seems at first glance, and intuitively, we understand this. Since all the complex life forms around have descended from one common ancestor, the chicken example shows once again that our biology can be easily controlled even by the most primitive selection, and to a much greater extent than we would like to think. After all, many people believe that man is the crown of nature, and only the sky and the stars are higher. But in fact, we have no reason to believe that humans are any different from chickens in this respect. We can either improve ourselves or make ourselves worse. Both can happen quite quickly by historical standards. This consideration is crucial, especially in light of the significant impact of adverse selection pressures in modern society, which I discussed in my video. A new era of humanity has begun. But the problem in happiness is that we can't just pick up and start breeding people with certain characteristics for a number of reasons. But even if societal morality allowed for such a thing, there remains the fundamental issue of timing. Human life cycles differ significantly from those of animals, such as chickens. For example, in developed countries, it takes an average of 28 years for a human to reach reproductive age from birth, while a chicken reaches reproductive age in just 6 months. This long human life cycle is the main reason why genetic changes in humans develop much more slowly than in rapidly reproducing animals, such as chickens. What if you found out that we could shorten the offspring cycle in humans to the same 6 months as chickens? Currently, with in vitro fertilization, IVF, parents can choose from a limited number of embryos, usually 10 to 15, because of the limited number of eggs that can be retrieved from a woman in a single procedure. Sperm count in men, on the other hand, can be said to be unlimited. However, if the number of eggs suitable for IVF is significantly increased, potentially by orders of magnitude, the pool of embryos available for selection can expand dramatically. We can't take an unlimited number of eggs from women, but we don't need to, because that's where stem cells can help us. If we can reprogram blood cells or skin cells into stem cells that will develop into fully developed eggs, the future of in vitro fertilization could change dramatically. Instead of retrieving and selecting a limited number of eggs, typically 10 to 15, women undergoing IVF could have hundreds or even thousands of eggs available for fertilization. This would allow prospective parents to screen and select embryos on a much larger scale, choosing those with the most desirable genetic traits. While this prospect may seem alarming, it is unlikely that humanity will abandon a technology with such enormous potential to eliminate devastating diseases, improve health, and increase life expectancy. Like other controversial technologies, such as explosives, nuclear energy, anabolic steroids, or plastic surgery, we often embrace advances that promise to improve our lives despite their ethical complexities. Embryo screening will initially aim to eradicate genetic diseases and, in the medium term, to improve other qualities, perhaps to prepare for life on a hotter Earth, in space, or on other planets in the future. This shift may eventually make natural conception unnecessary, as more people will opt for a safe and efficient procedure to select embryos with the most favorable genetic combinations. The decision to forego such technology and take a chance on natural conception will become increasingly rare as people prioritize passing on the most reliable genetic heritage to their children. How many people would be willing to watch a neighbor's child die from a preventable genetic disease and not blame the parents? In a world where embryos with genetic diseases, increased risk of cancer and diabetes, and predisposition to mental disorders are systematically screened out, the selection of embryos with traits associated with health and longevity becomes almost inevitable. For example, selecting embryos with genes associated with high IQ may become the norm, given their correlation with better life outcomes. Some might argue that predicting IQ based on genetics is a utopian fantasy, given the complexity and multitude of genes involved. Indeed, the genes responsible for IQ, like the genes affecting growth, are numerous. Some 800 different genes have now been identified that affect growth in a variety of ways. While this list is far from complete, Stephen Hsu, a theoretical physicist and vice provost for research at the University of Michigan, has demonstrated how future characteristics, such as height, can be predicted using known genetic factors. Hsu's team used data from 500,000 sequenced genomes from the UK's biobank to predict people's height based solely on their genetics. They compared these predictions to actual heights, finding an error margin of no more than 2.5 centimeters. This shows that we can estimate a person's height within a 2.5 centimeter range just from their DNA. However, predicting traits like intelligence is more complex. Intelligence is an abstract trait that's harder to measure directly. While this complexity doesn't make it impossible to predict, it does make it much more challenging. Currently, we've identified around 200 genes associated with above-average intelligence, but each gene contributes only a small fraction to the overall variation in human IQ. With only a few hundred genes known out of potentially thousands influencing intelligence, our understanding is still too limited to accurately gauge someone's mental abilities based solely on their genetic information. And yet the fact that we were able to find these genes so quickly at an early stage of genetic progress clearly points to our ability to find even more of them. Hans Bethe, Nobel Prize winner in physics, said, "I always thought that John von Neumann with his brain belonged to some other species, that it was a clear example of human evolution." And no wonder. Take a look at what and how much von Neumann is known for. American mathematician Hermann Goldstein said, "One of the remarkable features of von Neumann was his capacity for absolute recall. As far as I could tell, von Neumann was able, after reading a book or an article, to quote it verbatim. Moreover, he could do so years later without hesitation. He could also translate them without loss of speed from the original language into English. I once tested his abilities by asking him to remind me how A Tale of Two Cities begins, whereupon, without any pause, von Neumann immediately began reciting the first chapter and continued until about 10 or 15 minutes later, he was asked to stop." In their philosophical article, Embryo Selection for Cognitive Enhancement, Curiosity or Play, Oxford professors Carl Schulman and Nick Bostrom explore the possibilities of increasing intelligence through embryo selection. They estimate that the difference in IQ between the highest and lowest performing embryos in a typical IVF procedure involving 10 to 15 embryos is about 12 points. If you increase the number of embryos to 100 using stem cell technology, the difference could increase to about 20 points. Creating 1,000 embryos can increase this difference even further, to about 25 points. While a difference of 25 points may seem insignificant when 1,000 embryos are involved, it can mean a significantly different life experience. Still, that's not what you came here for. You're interested in the maximum IQ we can theoretically get. To do that, we'd have to do the unthinkable by crossing embryos with the necessary characteristics with each other. You heard me. Crossing human embryos with each other. The concept you are about to hear about may seem like science fiction or dystopia, but it is theoretically feasible. Jamie Metzl describes a process that is surprisingly simple. Imagine two parents undergoing IVF treatment with extensive genetic testing to select the embryo with the best genetic predisposition for high IQ. Instead of implanting that embryo, you let it develop for a while and then extract a few cells to create new sex cells, similar to how eggs are derived from the mother's blood cells. Now consider another group of parents who went through the same process but created sperm from their embryo rather than eggs. If we use the sperm from the second embryo to fertilize the eggs of the first embryo, these embryos become the biological parents of their offspring, and the father and mother become grandparents. These embryos, actually grandchildren, can then interbreed with embryos of other parents and ancestors, continuing this process indefinitely. The original grandparents will eventually become great-grandparents. The final product, an embryo resulting from several rounds of crossbreeding, is implanted in the mother to be carried to term. The not-so-pleasant nuance is that the child will be a descendant of its parents rather than their own baby. The idea of crossbreeding embryos to increase intelligence may seem radical, but some people may actually be interested in pursuing it. Over time, the process could drastically alter human genetics, potentially leading to people with unusually high IQs. By crossbreeding embryos, instead of 3.5 generations in 100 years, we could change 200. Stepping backwards by the same number of generations would take us back to when the wheel was invented. By crossing embryos with selected smart genes, we could create intellectually exceptional humans. So what level of IQ could we theoretically achieve using this method? Shulman and Bostrom calculated that crossing embryos in 5 generations, selected from 10 different groups based on their highest IQ, could result in an average IQ increase of about 65 points. To put this in perspective, the average IQ is 100. If you add 65 points, the IQ would be 165, an unusually high level of intelligence. By comparison, Einstein was rumored to have had an IQ of 160, but there is no concrete evidence for this claim. Nevertheless, a 65-point increase in IQ would be significant. There is a difference of those same 65 points between ordinary people and notional geniuses. That is, the difference between a conventional genius and an ordinary person is the same as between an ordinary person and a person with a severe form of mental retardation who requires constant care. A 65-point IQ increase, achieved in just 5 generations, represents a significant leap in intelligence. Extending this process to 10 generations, as Shulman and Bostrom suggest, could result in an astonishing 130-point increase. To put this into perspective, a 130-point increase would take you from the average IQ of 100 to a level comparable to Terence Tao, whose IQ is reported to be around 230. Tao is often described as the mathematical Mozart due to his exceptional abilities. For those not deeply familiar with advanced mathematics, Tao's achievements, such as his probabilistic proof of the strengthening of Shemarati lemma and his work on the circular law in random matrix theory, might sound like a jumble of complex terms. What stands out, however, is that he was already taking university-level math courses at the age of 9. Here's a quote from Princeton University math professor Charles Pfefferman from a New Scientist article about his abilities. Tao's reputation is such that mathematicians now compete for his attention to their problems. He is becoming something of a lifeline for jaded researchers. If you are stuck on a problem, the only way out is to get Terence Tao interested. An IQ of 230 is already an astonishingly high level, seemingly unattainable for most people. However, Stephen Hsu, an American physicist known for his work on genotype-based predictions, is even more optimistic. Hsu predicts that within the next decade, we will be able to discover most of the genes that affect intelligence. This could open the way to creating people with unusually high IQs, potentially around 1,000. While it is hard to imagine what an IQ of 1,000 might mean, it is clear that such a level would surpass the cognitive abilities of all humans who have ever lived. This is of great significance and suggests that human intelligence may reach unprecedented heights in the future. Hsu writes, "We can expect people with extraordinary mental abilities that can manifest simultaneously and at full capacity. Perfect memorization of language and images, ultra-fast thinking, and computational ability. The most vivid visualization of geometric space and other dimensions, the ability for multiple analysis, parallel thinking, and the list goes on." While the prospect of creating humans with an IQ of 1,000 is intriguing, it comes with significant risks and uncertainty. Evolution has endowed us with a range of defense mechanisms, which means that a person with such an unusually high IQ could face unforeseen problems such as mental instability, psychosis, or neurological disorders. In addition, another layer of complexity is added by the possibility of harmful mutations arising from embryo crossbreeding. And of course, the creation of super-intelligent humans will significantly affect social and ethical aspects. As for the technology, it may seem far-fetched, but it is already advancing. Recent advances include Chinese scientists successfully obtaining sperm from stem cells taken from a mouse egg, which were then used to fertilize the egg of another female mouse, resulting in healthy offspring. Similarly, American scientists obtained viable offspring from 2 male mice. Although there is still a long way to go before these technologies can be used on humans, these developments show that the principle itself is workable. This suggests that same-sex couples can have biological children, demonstrating the technology's potential for future applications. Particularly interesting is the ability to obtain an egg and sperm from the same person, making them both the mother and father of their child. Despite the seemingly bizarre nature of these advances in human reproduction, any child born this way will still have all natural human DNA, untouched by gene editing technologies such as CRISPR. Genetically, despite the diversity in appearance, languages, and cultures, humans are remarkably similar. As a study led by the University of Oxford on a group of chimpanzees in Central Africa has shown, they are more genetically different than people living on different continents. One reason is the fact that many times throughout history, human ancestors have been on the brink of extinction. Genetic studies show that about 1.2 million years ago, our ancestors were reduced to just 26,000 individuals. Then, about 150,000 years ago, the human population may have fallen to about only 600 people, balancing on the brink of survival in southern Africa, which is comparable to the size of a small village. And then again, when 70,000 years ago, ash from a Sumatran volcano covered the sun for 6 years, bringing the human population down to just a few thousand individuals. For evolution, 70,000 years is a relatively short time, given its slow and conservative nature. Direct genome editing technologies, such as CRISPR-Cas9, carry significant risks. Even if we perfect the ability to precisely cut and alter genetic sequences, we could still disrupt genes whose roles are not fully understood, leading to potential long-term consequences that may not manifest immediately. These effects may not manifest until later in life or in future generations. This is why companies like Deep Genomics are using artificial intelligence alongside genomics to predict disease and understand genetic function. Their approach aims to address the complexity of biology, which often exceeds human cognition. You see what the problem is. We don't have, and are unlikely to have soon, an understanding of what each specific gene is responsible for. Currently, our understanding is limited to observing correlations between genes and traits in large samples. For example, we take the genomes of people with schizophrenia and compare them to a large sample of genomes of healthy people of the same sex, age, and so on. And we look at exactly where they differ. It is very difficult to understand all the functions of each gene. It is very difficult to understand its relationship with other genes. As scientists delve deeper and deeper into genetics, the concept of a gene has become increasingly complex and abstract. Richard Young, a geneticist at the Whitehead Institute, notes that what used to be a simple explanation of genes now requires months of lectures to fully articulate. This shift is not due to a decline in student understanding, but reflects the growing complexity of genetic science. So, we don't know how we can make significant gene modifications to complex traits without deliberately harming the rest of the genome. But if it is all only very complex rather than infinitely complex, there will come a time when the complexity of our tools will surpass the complexity of our biology. On the other hand, of course, no one will wait for that. The very idea of altering our genes requires an extreme degree of judgment. But we'd be a very different species if our thoughts were guided by judgment rather than ambition. Just as we did not have perfect knowledge of fire until man began to use it, of cancer until we tried to cure it, or of home crops until we began to eat them. In every instance of human development, we have found a balance between benefits and risks, relying on imperfect knowledge in the pursuit of discovery. Genetic technology will be no different. We know for sure from existing examples in nature that genetic modification can give us biological immortality like a hydra, eyesight like an eagle, strength like gorillas, or the ability to see in 16 dimensions like the mantis crayfish I discussed in the video, the invisible infinities, and many more things we don't even realize we have. These examples highlight the potential for genetic technology to unlock unusual abilities. While some may argue that genetic modification is unethical or that we cannot make decisions for an unborn child, it is already part of our reality. For example, in 2006, the Korean government imposed a 10 p.m. curfew on intensive schools due to concerns that children were becoming overworked and exhausted from studying late into the night. Despite these efforts to reduce pressure, the educational arms race in Korea continues. Physical beauty is valued around the world, but Korea has taken the beauty cult to a whole new level. It is estimated that South Korea has one of the highest per capita plastic surgery rates in the world, with a large percentage of women in their 20s and 30s undergoing procedures. This cultural emphasis on beauty reflects a broader societal trend where people are willing to go to great lengths to achieve desired results. Korean parents often give plastic surgery to their children as a graduation gift. Here's a quote from a Korean college student interviewed by The New Yorker. "All girls get plastic surgery at 19. So if you don't get it done, in a few years all your friends will look better and you'll look like a worse copy of yourself. Do you think this country won't have genetic modification as soon as it becomes available? Do you really think so? And how will your child look at you when they realize that you could have made them smarter, taller, prettier, but you didn't? Even if you try your best to adhere to the principle of naturalness for your offspring, it still won't help, because people with better genetic characteristics are bound to have more sexual success and consequently more offspring. Consider the example of blue-eyed people. All blue-eyed people share a common ancestor who lived about 8,000 years ago and whose genetic traits proved to be quite successful. The prevailing theory is that blue eyes became an attractive trait that influenced mate selection. Now imagine a future in which your natural descendants are surrounded by exceptionally beautiful and intelligent people. In such a scenario, despite improvements, these offspring may have difficulty with reproductive success if their traits are perceived as less desirable than the improved norm. After a few generations, this could reduce their chances of passing on their genes. But in the long run, it's not that big of a deal. Consider this. The more genetic modifications you put into your child, the less your child will be your child. Genetically modified children will not have parents. They will have designers. The Genome Recording Project is a groundbreaking initiative that aims to raise $100 billion to synthesize the complete human genome. Starting with the simplest of organisms, the project plans to move on to the creation of the entire human genetic code. This project represents a significant leap from CRISPR technology. While CRISPR allows for editing existing genetic material, Genome Project Write aims to create life from scratch, which is akin to writing a book rather than simply editing it. We will be able to literally create life. In the 1997 movie Gattaca, the plot centers on a genetically unmodified man who aspires to become an astronaut, is rejected based on his genetic profile, but he eventually succeeds through sheer determination and ingenuity. In reality, however, society may not want to send a genetically unmodified person into spaceflight when modified humans can offer benefits such as increased resistance to radiation and better bone density and weightlessness. Striving for equality is important, but absolute equality is not always possible or desirable. For example, while villagers in Bangladesh now have affordable smartphones thanks to the industry, insisting on equal access from the start could have stalled the industry's development. Similarly, Inscripta is working to democratize CRISPR technology, making its tools readily available and potentially accelerating progress in genetic engineering. The idea of turning biology into a computing platform where DNA is code and CRISPR is a programming language reflects a vision in which genetic engineering becomes as fundamental and transformative as computer science. As Wired magazine noted in May 2018, this vision envisions a future in which we can manipulate biological systems with the precision and creativity of programming. Our desire to play God through technology can be attributed to a sense of human superiority and a desire to overcome what Charles Darwin called the clumsiness and cruelty of nature. This desire drives us to innovate and push the boundaries of what is possible. If technology can be created, it will be created. It is we, not someone else, who will do it first. Speaking of which, China, in particular, has embarked on the most aggressive path of creating its own gene pool on an industrial scale. The 13th Five-Year Biotechnology Industry Development Plan, developed by China's National Development and Reform Commission, aimed to obtain the genomes of at least 50% of all newborns by 2020 and support hundreds of individual genome sequencing and clinical data collection projects involving private companies and local governments. China is also working to standardize electronic medical records across the country and ensure that privacy measures do not impede access to information by researchers, companies, and the government. Interpreting the vast amounts of genomic data generated today is a challenge beyond the capabilities of even the most advanced computers. As genomic datasets grow, the potential of supercomputers and artificial intelligence becomes crucial to unraveling complex genetic patterns and understanding their implications, as AI is expected to play a key role in deciphering the genome. China's strategic drive to take the lead in AI innovation will have a significant impact on the future of human genetic engineering. Beijing's ambitious AI development plan, released in July 2017, envisions China becoming the global center of AI innovation by 2030. China's rapid progress in genetics is due in part to a vast and largely unregulated gray market of human genetic engineering, and not surprisingly, it is where genetically edited humans were first born. As I detailed in the video, a new era of humanity has begun. Now imagine the scale of the problem. Imagine what's at stake. Imagine two groups of people in different political and social realities. For example, the European Union has taken significant steps to protect individual rights. The General Data Protection Regulation, which came into force in May 2018, requires the explicit consent of every EU citizen before their genetic data can be included in any database or research. This regulation reflects the EU's commitment to protecting personal data and ensuring ethical standards in genetic research. China's approach to genetic data collection stands in stark contrast to that of the European Union. While the EU focuses on protecting individual rights and preventing the misuse of genetic data, China's Ministry of Public Security is building the world's largest DNA database, giving the state extensive control over genetic and other personal data collected within its borders. The power and impact of genetic research is closely tied to the availability of large and high-quality datasets. The larger and more complete these datasets are, the faster we can uncover the genetic underpinnings of complex diseases. The organization with the most extensive and detailed datasets is likely to lead the genetic revolution and reap significant benefits in the form of wealth, prestige, and influence. This raises some questions. For example, what can we do? We can try to persuade the world not to genetically modify. But first, many societies, as we have seen, are running enthusiastically toward the age of genetics. And second, although virtually all world leaders agree that man-made climate change could make our planet uninhabitable, we couldn't even get a global convention to at least try to stop the warming. So how do we stop the spread of modified genes, given that all genetic changes will be inherited? Remember the blue-eyed people. Should we impose travel restrictions on China or ban Chinese citizens from traveling abroad? Would you support the use of military force to prevent genetic changes in humanity? Imagine that a powerful country like China became a leader in introducing advanced genetic and other technologies to improve its population, and another country, such as the United States, decided to abandon these advances for political, ethical, or other reasons. What awaits us 100 years from now? It seems likely that societies that reject genetic modification technologies will quickly fall into oblivion, despite the problems and setbacks that may be encountered by those who embrace the technology. It is therefore unlikely that gene editing will be purposefully abandoned. On the contrary, the freest countries, where gene editing will not be monopolized by the state, are likely to dominate the future market for gene editing technologies. As Jamie Metzl writes, companies and countries that manage to crack the code of diseases such as cancer and a host of others, and learn to understand and potentially modify other human traits, will become more than just a biological version of Google or Alibaba. They will be comparable in power and influence on the Mongols of the 13th century, the British of the 19th century, and the Americans of the 21st century. Of course, this scenario comes with significant risk. Look at how quickly we have come to the absolute leadership of specific companies and the total uniformity of their products. If everyone starts choosing the same genes for their genetically edited children, it could lead to a dramatic reduction in genetic diversity. Such uniformity could make populations more susceptible to disease and reduce our overall adaptability. This would expose us to as yet unknown risks. Remember the corn we domesticated over 8,000 years? Well, it has become so genetically homogeneous that its susceptibility to disease has increased dramatically. And I think Christopher Nolan had a whole movie about it. If a new virus or pathogen emerges, the lack of diverse genetic traits could mean that there are no individuals with beneficial mutations to survive and counter the threat. And this is only what we can conceive of because we are completely unaware of the evolutionary advantages of our diversity in various aspects. If humans had the ability to edit genes in prehistoric times, selective pressure could have resulted in the elimination of traits that seem unfavorable today, such as those associated with autism. However, such traits could also provide unexpected benefits. For example, a person with autism could make a groundbreaking contribution to Facebook. Conserving genetic diversity is crucial because it includes a range of traits that can be useful in unforeseen circumstances. Evolution acts conservatively, modifying existing traits rather than introducing entirely new ones. It builds on what already exists, maintaining a balance between preserving basic traits and adapting to new conditions. This conservatism ensures that when new traits are introduced, the fundamental genetic structure remains stable. This is the reason why the skeletons of bat wings and human hands and whale fins are so remarkably similar. They even have the same number of bones. This indicates that evolution builds on existing structures rather than creating entirely new ones from scratch. Our understanding of human behavior and personality may seem far removed from our primitive ancestors, but in fact many of our psychological traits are strongly influenced by genetics, Although eugenics is often criticized as unethical, it is important to recognize that forms of human selection have been part of society for centuries. Historical practices such as infanticide, noted in Roman law, reflect the selective pressures that have influenced human populations throughout history. These practices, although severe, demonstrate that human societies have long been involved in shaping their genetic futures, albeit unintentionally. It was widely practiced in the Roman Empire. The fourth of the laws of the 12 tables of Roman law stated the following: "A father shall forthwith put to death a son characterized by ugliness or having a form distinct from the human race." In ancient Sparta, city elders personally screened newborns to see that no sick child survived. German tribes, pre-Islamic Arabs, ancient Japanese, Chinese, and Indians all practiced infanticide. Today, although such practices are no longer common, modern societies still influence genetic outcomes in more subtle ways. Laws and societal norms affect reproductive success indirectly. People with a predisposition toward cruelty are likely to go to prison, leading them to leave fewer copies of their genes on average. In this sense, modern human populations are shaped not only by cultural patterns, but also by the selective pressures exerted by our social and legal systems. We think of humans as very rational beings with critical thinking, but evolution didn't select us along those lines. Even if you don't belong to any of the existing religions, if you consider yourself the most unbelieving person in anything, it still means absolutely nothing. Because for the most part, what you believe in doesn't feel like faith to you. You just think the world is the way it is. Paul Rosen of the University of Pennsylvania is one of the pioneers in the study of magical thinking. He believes that with the advent of the mind, the primary emotion inherited from animal ancestors expanded dramatically to include, among other things, the idea of contact, the transfer of filth by touch. For example, volunteers in Rosen's experiments flatly refused to drink juice touched by a sterilized cockroach or to eat from an immaculately clean chamber pot. Many people can bring themselves to drink juice or eat from a pot, but something inside us will resist it. What is it? That's it, the result of evolution. We think truth is what corresponds to objective reality. But for your genes, that's not true. For your genes, there is only one truth. For them, truth is what allows them to pass on their copies to the next generation. No objective reality is of any concern to them. Those primates that were not particularly squeamish did not leave their genes behind. Those primates that didn't feel discomfort and the urge to scratch from a mere cluster of holes didn't leave their genes behind. Those primates that did not pause at the sight of a snake-like stick did not leave their genes behind. Those primates that did not experience the strange pleasure of a babbling river, and consequently, the desire to be near it, did not leave their genes. Those primates who were not drawn to stare at the fire for no reason, and consequently, to stay close to it, did not leave their genes. You experience a kind of rejection from people with 6 fingers, not because you have decided so yourself, but because your genes are set up to choose 5-toed relatives. There is nothing objective or rational about that. This list could go on for longer than you'd be willing to listen to. We are rarely aware that much of what we do is not an act of our free will, just as we are aware that the existence of such a thing as free will has yet to be proven. It is hard to imagine how much of our behavior is not the product of our conscious activity. How confident can you be that a person's personality is completely disconnected from their biology, given that your face alone can predict your political orientation with 72% accuracy? For 4 billion years, our genes have been culled by the environment in a slow and painful way. That's why we're so adapted to that environment. We are entirely its product. And now we're about to edit our genome. How sensible is it to delegate the selection of our genes to companies or states that, unlike the environment, will be able to not just favor some genes and discourage others, but change them instantly, and do it still through a black box, aka artificial intelligence? It's a slippery slope. Will it not lead to the fact that people, generation after generation, will begin to genetically adapt, including to the policies of those states and companies that select them with the unintentional or intentional assistance of the latter. Imagine people absolutely loyal to particular parties, so loyal that it will be impossible to change their opinion at the level of physiology. Attempting to show them the negative aspects of their political regime would make them fearful, dizzy, disgusted, and nauseous. Or imagine that you own a company that provides genome editing services to the public and that has captured a large share of the market. And such a company is sure to come along sooner or later. Your product is literally your potential customers. You are projecting them in huge numbers. Wouldn't you be tempted to use genome manipulation to add to your company's prospects? But even if not, consider this. What is the main criterion for selecting genes? If the focus of genetic enhancement were to maximize individual happiness, it's possible that over generations, humans might become increasingly content with their circumstances, regardless of any underlying issues or limitations. This could result in a population that is physiologically and psychologically satisfied with the status quo, potentially to the detriment of critical thinking and innovation. Such a scenario might limit our ability to recognize and address problems or changes in our environment, leading to a kind of self-imposed stagnation. It's an important consideration for policymakers and researchers, as focusing solely on enhancing happiness or IQ could unintentionally narrow our capacity for adaptation and problem-solving. The Pentagon and other strategic organizations might indeed benefit from contemplating these potential outcomes. Understanding the broader implications of genetic and psychological modifications on human adaptability and awareness could be crucial for long-term planning and decision-making. Thank you for watching. Special thanks to the sponsors. If you want to support my channel, put a like and subscribe.